Okay, we're back again. Another Year 12 biology lesson. This time we're going to focus on the topic of meiosis. And as we always do, we'll start with a little bit of knowledge retrieval. So we'll go back to mitosis. On the screen, you've got a number of kind of letters and numbers. And what I want you to do is pause the screen, and then can you work out what a logical order for those letters might be? And can you then jot down a description of what's happening at each stage? So pause the video now. Okay, hopefully you've had the chance to do that. So now you can go through and see if you've got them right. So let's have a look. So as we work our way through, the I part is obviously looking at interphase, and this is looking at being composed of G1, S and G2. So we'll look at those individual components now. So G1, we're looking at growth of the cell and production of proteins for cell division. So that's called gap 1. As I said before, I prefer the word growth, it's more descriptive of the process. We're then moving on to S. S is when we've got our DNA synthesis, our semi-conservative replication of DNA. We then have G2, and G2 further growth and replication of the organelles ready for the actual cell division process. As we move on from G2, that's kind of the end of interphase. We're now getting into the mitotic phase. The first bit we're going to look at is prophase. Prophase is we're looking at chromosomes becoming visible. The reason for that is they're becoming shorter and they're condensing. The nuclear membrane is now breaking down too. Metaphase, things go into the middle, from a metaphase to the middle. So our chromosomes are going to line up in pairs at what's known as the equator of the cell. Spindle fibers are going to start to form from the centrioles, the edges of the cell, and attach to the centromeres of our chromosomes. And on to anaphase. Anaphase, we're then going to move the sister chromatids, are going to be pulled to opposite poles of the cell by the spindle fibers. Remember careful with the language here. I would avoid saying things like the chromosomes are pulled apart because it's somewhat ambiguous. Focus on the sister chromatids being pulled to opposite poles of the cell. So that's I, P, M, A, and then we're looking at T, moving on to telophase, and the nuclei are going to reform at this stage. The cell membrane will ultimately start to fold inwards and will end the process as we move into cytokinesis. So that's a recap of mitosis. So let's move on and have a look at the process of meiosis. Okay, be careful with spelling. Just make sure that if you are making note of mitosis and meiosis, that it's clearly which one you're referring to. It is something an examiner could pick up if, again, it's slightly ambiguous which form you're referring to. Okay, so it's important that we get some of the definitions right within this next section. So we'll look at the term haploid, a term describing a cell with only one copy of each chromosome. It's usually the sex cells. There are exceptions. We'll come to that later. N, the single letter N, is used to denote haploid. And diploid, we use the term 2N because it has twice the number of chromosomes as the haploid cell. This is usually found in the body cells, also known as the somatic cells. Just got the picture at the bottom. I'll just kind of zoom into this, so bear with me. Let's have a good look. So this is what we usually call our homologous chromosomes here. Okay, we've got our single chromosome with a centromere structure. As we go through DNA synthesis, we go through semi-conservative replication to produce this kind of structure. Frustratingly, I think for many students, we call each of these structures on this side a chromosome. Once we've formed this form, where we've got two sister chromatids held together at the centromere, we also call this a chromosome. So it's sometimes worth kind of adding an extra little bit of information if you're referring to this form, where the cell is just about to divide. I'd always try and recommend including the term composed of two sister chromatids held together at the centromere, so it's obvious what you're referring to. Okay, so let's move on a little bit. And kind of just summarize the key principles of what's going to happen. So during meiosis, we're looking at homologous pairs of chromosomes. So that's kind of one from each parent. And we're going to separate those. So we've only got one chromosome from each parent entering the gamete that is formed. we'll end up producing 4 daughter cells, and each of these daughter cells would have half the DNA that the original cell started with. And that can then lead to, in sexual reproduction, we can then get our particular gametes are able to fuse together to give rise to the new offspring. And this is going to require 2 haploid cells to come together. If we have 2 haploid cells forming, joining together, that would lead to us getting a single diploid cell, which can then go through mitosis and form the adult organism. Just a reminder again about that kind of terminology, going from homologous chromosomes. So, key thing about meiosis, it's a process that's only found in organisms that undergo sexual reproduction. It leads to production of haploid gametes, eggs and sperm, pollen, ovule, for example. It takes place in the sex organs, so we're going to ovary, testes, and implants the stamens and carpel. And the whole purpose of meiosis is to introduce genetic variation into the next generation. And we'll look at how that occurs as we move through the rest of this presentation. So let's have a look at an example where meiosis doesn't occur, because that should hopefully help illustrate why meiosis is important. So if we've got the image on the left hand side shows the X and Y chromosomes from the male, and we've got 2 X chromosomes from the female. We're not looking at all 46, that would make the image somewhat confusing, but we're looking at adults that are diploid, and if they didn't go through meiosis, the sex cells that they would produce would have the same number of chromosomes as the adult cell. In this case it would be a diploid number, and a diploid number in the egg. So if we had fertilization occurring here, that would lead to the zygote forming that would have 4N, which would be an organism or a cell that will be non-viable. So what we need to look at is what the process would be if we have meiosis. So with meiosis, that leads to our half set of chromosomes. So we're going from our diploid number at the top here, we're going through a process to reduce that diploid number to a haploid number. And the same is going to occur with the egg cells. So we've now got sperm cells that have a haploid number, we've got egg cells that have a haploid number, and if any of those sperm cells fertilize any particular egg, you can see we combine those together, we're going to end up back with the diploid set. So one of the key purposes of meiosis is to produce a haploid cell, so that upon fertilization we're then back to a diploid cell, back to that 2N number of chromosomes. So essentially meiosis actually has 2 divisions, unlike mitosis. So the first meiosis stage is sometimes called meiosis 1 and meiosis 2. And then we have the anaphase, prophase, metaphase, and so on, divisions within each one of those. So meiosis 1 is all about separating out the homologous pair of chromosomes. So here at the left we can see the 2 chromosomes that have gone through sort of semi-conservative replication to produce the 2 sister chromatids joined at the centromere. We've got 2 chromosomes, so the first stage is about separating those pairs out. And then meiosis 2 is almost the same as mitosis, if you like. It's about moving the sister chromatids from whatever chromosomes are in that cell to opposite poles of the cell, and then forming 2 new cells beyond that. So you can see in the image here we've now got a single, what was an original sister chromatid. It's been separated from its other sister chromatid. So this meiosis 2 is very similar to mitosis. So, key process. During interphase, each chromosome is going to replicate, and each chromosome is going to be made of 2 sister chromatids. So the first bit of meiosis is exactly the same as mitosis. We have to have a DNA replication stage. So we end up with that structure of, we're more familiar with, that X-shaped chromosome made from 2 sister chromatids. These are meiosis 1. We then have our homologous chromosomes kind of come together, and when they come together and they kind of wrap around each other, or some of them do, and at that stage we can describe that pair of homologous chromosomes as a bivalent. Okay, a bivalent. So that is composed of 2 homologous chromosomes. So you would have if you like, 4 chromatids within that structure. And when that happens, you can get the formation of a structure called a chiasma, or if you like in plural, a chiasmata. And these chiasma are where you get an exchange of genetic material between different chromosomes. And we get this crossing over occur, and that would, because it exchanges genetic material, that's bringing in genetic variation. We also then have in a process where those chromosomes, what's known as independently segregate or independently sort. So the maternal and paternal chromosomes that we have can move independently to the opposite poles of the cell. And again, we'll look at that in more detail in a minute. And then meiosis 2, we've already said, almost the same as mitosis. That's where the individual chromatids that make up that cell can then separate. So let's have a look at this as an animation. It's always easy to visualize this process when you can see things moving. So here we have an image of a cell that has already gone through DNA replication. We know that because we can see the individual sister chromatids. So these are going to condense so they become a little bit more visible, so we can kind of see a bit more space to kind of see the arrangement. And we see the centromere in the middle. The nuclear membrane has broken down. I'm not concerned about calling these stages prophase, anaphase, telophase. You will see that in some textbooks and some revision sites. It's not something we need to concern ourselves with. We need to sort of describe the process and be able to explain what's occurring. So here we have our chromosomes with 2 sister chromatids, and the next stage is where these ones are going to kind of come together. So we've got our homologous chromosomes, so these particular colours, the red and blue, denote from an original parent. So maybe the blue one was the paternal chromosome, the red one was the maternal chromosome, but they're going to come together and form our bivalent. Okay, so now we have these 2 homologous chromosomes coming together, made of 2 sister chromatids each. We've now got 4 chromatids in total. This is our bivalent structure. And at this stage, you can notice at the bottom down here, I'll just zoom in briefly. Okay, notice at the bottom down here, we've got this opportunity here where we've got these sister chromatids kind of coming into contact with each other. Okay, so that particular stage, you can see where these 2 sister chromatids are coming close together. That's how we can get the genetic exchange of material. So we'll zoom in and have a look. So we can just get a bit of a break in the chromatid, and one part of one chromatid would be exchanged with the other. So once we've done this stage, you can see now why that would lead to some genetic variation, because the 4 chromatids that we have are now different. One chromatid was originally from totally from the paternal line. The next chromatid has now got part paternal with a little bit of maternal mixed in. The next chromatid's mainly maternal with a bit of paternal, and so on. So that crossing over stage is important in terms of introducing variation within the chromatids. Remember, in human cells, we're looking at 23 pairs of chromosomes, so any of the 23 can undergo this particular process. It's a random event. It can occur anywhere. It doesn't need to just be on the ends, although in most textbooks that's how it's shown. So you can bring about a huge amount of variation with that particular process. So once the crossing over part has taken place, we then go to separate out and move apart those particular homologous chromosomes to opposite cells. And then we're going to go through the second stage of meiosis. Again, this is very similar to mitosis. Whereas in the second stage, we're then going to separate the individual chromatids, and they're going to get pulled to opposite poles of the cell, just like we had in mitosis. And we've now got 4 daughter cells, each one of them now haploid. We've got a half set of chromosomes. We started with a cell that had 2 chromosomes. We now have a cell that has 1. And if you look really closely at the image, you can see that each one of these individual chromosomes represented in these pictures is unique. This one has come from I said paternal, I think was the blue, and we said maternal for the red. And looking at these ones down here, this one is mainly all from the original paternal chromosome. It's got a little bit extra that came from the maternal one, and this one vice versa. So that has brought about 4 unique gametes produced from that particular division. Remember, we're looking at a single chromosome. think about a human, we're looking at 23 pairs of chromosomes, so we've then got, again, a lot more variation that can be introduced. So what about independent segregation or independent assortment? This is kind of thinking about when they're lining up at that original stage, and we're about to get crossing over, and it's being aware of where those original chromosomes come from. So I'm going to use yellow for maternal and red for paternal. So if they're going to line up in their homologous pairs like such, okay, so we've got this particular pair lining up here, and we'd then come together, we'd go through crossing over and so on. We're just going to look at what would happen when these homologous chromosomes get pulled to opposite poles of the cell. So it may be, quite simply, that this occurs. Okay, so all of the maternal ones got kind of pulled to form one new cell, and all of the paternal ones got pulled to form another new cell. And that would lead to that kind of formation when the gametes are formed. These aren't showing crossing over, we're just looking at independent segregation, but that's kind of one way how these particular cells down this side over here could have separated. But it didn't have to be like that. It could have been like this, for example. Again, we've got maternal, paternal, because how they line up at the equator is an independent process. Each pair of homologous chromosomes can pair up independent of the next pair, independent of the next pair. And it's just how these individual pairs segregate. So it may be that they're segregated like this. So again, you can see quite quickly in this particular cell, I've now got two that were originally maternal and one paternal, and here two paternal and one maternal. And we can therefore see that the gametes that were produced are different. Look at the different combinations that you can get. We've got something of the order of, if we have in humans 23 pairs of chromosomes, I just wonder if you can figure out how many different possible combinations of those chromosomes you can actually get when you get to this particular stage. So just pause the video, have a think about that. Okay, we've got in humans 23 chromosomes, 23 pairs of chromosomes I should say, and if each of them can independently segregate, okay, independently assort, how many different combinations can that lead to? So pause the video, have a think, and I'll give you the answer in a minute. Okay, now time for the big reveal. In any diploid species, what we're looking at in terms of the maths to work out the possible combinations from independent assortment is a relatively straightforward calculation. We've got 2 to the power n, okay, where n is the number of chromosomes that we can get. So for humans, that comes out to be 2 to the power 23. Okay, if we then do that calculation, 2 to the power 23, that's going to lead to something of the order, I'm just learning how to use my tablet here, 8,388,608 possible combinations in terms of independent assortment. So you can see straight away the degree of variation that would produce. If you bring that in and kind of look at that as well as looking at crossing over, ultimately you've got an infinite number of possible combinations. So for humans, 2 to the 23 over nearly 8.5 million different possible combinations of those chromosomes coming together, you can see how this process can lead to such vast variation. So let's focus on the crossing over stage, okay, in a bit more detail. So here we've got our bivalent, if you like, the two homologous chromosomes coming together, and you get kind of a point where a couple of sister chromatids from the maternal and paternal chromosomes kind of come close together and form a single chiasma, or if you've got lots occurring, you've got many chiasmata. And in that process, because of that, we get kind of a break that occurs and an exchange of genetic material. So there could be many genes in this particular sequence that have now been kind of swapped from one chromatid to another. And we've got our recombinant chromosome that's formed. So these individual chromatids that we can see here, each one of them is different. This one came from one parent originally, this one from the other, and we've now got ones that are composed of two separate little bits. So let's kind of think about that in a slightly bigger in more detail in terms of what the impact might be on particular genes. So here's an example. We've got an original chromosome where we're just looking at genes B and G. I'm using capital letters and lowercase letters just to denote different versions of those genes. So I've got different alleles. Remember that from GCSE, the term allele, different version of a gene. So this particular chromatid has, in this case, the dominant allele B and the dominant allele G, and this one here has the recessive forms. So if we go through that process with meiosis, what that would lead to is quite simply we would replicate the DNA. So we'd get an identical chromatid to the one we started with. This is my centromere holding those two sister chromatids together. So this chromatid is identical to that chromatid. This is my whole chromosome structure, and the same with the other chromosome structure. But if I then go through a process where I've got crossing over that's occurring, okay, and if you look kind of down at the bottom where I've got the particular G gene and those particular alleles that we've got, you can see if I exchange particular components down here and swap some of that genetic material over, so that's going to lead to this particular combination. So I've exchanged genetic material, so there's my original chromatid. I've got one of the other original chromatids, but on this particular one, this one has that particular allele with the dominant B allele at the top there, and going down here at the bottom, we've got the recessive form of the G gene, and originally it was this one, so we've exchanged and moved alleles from one chromatid to another, so you can see how that would bring about particular variation in the offspring. And all gametes will have a different combinations of alleles. So each one of these particular gametes that we've got here, just looking at that particular chromosome, each one of them is different. Remember humans, we're looking at 23 pairs, so we can start thinking about the possible combinations. Crossing over can occur anywhere. It's a random process, so we've got a huge degree of variation. Just to summarize really quickly, so we've got originally going through the process, we start with our interphase where we replicate the DNA, semi-conservative replication. We then have our homologous chromosomes that would pair up and undergo some form of crossing over. And at the end of that process, we would then independently segregate or independently assort those particular chromosomes, so that in the end we have this kind of structure where we've got 4 daughter cells, and each daughter cell has a haploid number of chromosomes. We've also got a degree of variation because of independent assortment, the maternal and paternal chromosomes separating, and we've got our crossing over. Very easy to think about meiosis and mitosis with kind of the human hat on, if you like. There's kind of lots of misconceptions about all organisms have the same kind of life cycle that we do. So just looking carefully at this image, you can see that this represents human life cycle. So we've got the meiosis phase producing our eggs and sperm. Our haploid gametes then have fertilization to produce a diploid zygote, that original fertilized egg cell. That will go through mitosis and differentiation to produce our adult organism that can then, in the sex organs, can then go through meiosis again, producing haploid gametes, and the cycle will just continue. All organisms are the same. We do have some differences, and it's being able to kind of spot that, spot what's different in different organisms. So here's another organism. Okay, we're looking at an example of some particular plants here. So if we go from a fertilization stage, we've got our diploid zygote, just as we had in humans. We've then got mitosis to produce the adult organism, you can see down here. And that would be a multicellular organism. It's got diploid cells. And then it could go through meiosis. In this case, it's going to produce structures called spores. These spores are haploid. So we have gone through our meiosis phase. We have introduced genetic variation into these particular spores, but those spores can undergo mitosis. Okay, so mitosis is about making identical cells. So those spores can undergo mitosis, and you can produce a multicellular organism that is composed of haploid cells. This multicellular organism can then produce gametes. Those gametes can fuse together to bring about fertilization again back to a zygote. So the difference with humans is we've got a multicellular haploid organism here and a multicellular diploid organism, whereas with humans we don't. We've got quite simply that production of, that production of haploid gametes and a diploid multicellular organism. So you do get some differences. Okay, one example is probably worth being aware of is looking at bees. Probably aware we have a queen bee. So if we look down the bottom here, we've got a queen bee who is diploid, okay, 2N, and the queen bee can undergo meiosis to produce egg cells. Egg cells would be haploid. And what can happen with those egg cells, okay, if we kind of follow the thread down at the bottom of the screen down here, what can happen with those egg cells can undergo a process called parthenogenesis. Now, parthenogenesis is where you can produce an adult organism where you've had no fertilization. So in this case, that parthenogenesis process from that individual egg cell produces what's known as our drone. And our drone is effectively a male, okay, it's a male bee, our drone. And that drone bee, if we kind of move further back up here, that drone bee can still produce its own sex cells, its own sperm cells. It can't be via meiosis because it's only haploid to begin with, so it has to produce the sex cells through mitosis, so they're all clones. That sperm cell that's produced can be used in fertilization to fertilize some of the egg cells that were produced by the queen. So the sperm cell can fertilize an egg cell, and after fertilization we're back with a diploid organism, a diploid female. If the diploid female just eats honey, that diploid female would develop into a worker bee, still 2N, and that worker bee would be sterile. However, in the hive, some of those bees can be fed what's known as royal jelly, which you've probably heard of, and if some of those newly developing bees are fed royal jelly, they can develop into a queen. The queen is still a diploid, but that queen is not sterile. That queen can produce egg cells, and from fertilization we can kind of complete and carry on the process. So it's being aware of not every life cycle is the same as humans, and can you sort of spot any differences when you're given something slightly unusual. Classic example of apply what you know to something you're unfamiliar. Okay, it's kind of one of your assessment objectives. So let's move on and have a look at the kind of exam questions that you're likely to get. So I'm going to let you kind of have a read through the question, trying to think what you would do for an answer, and then we'll go through and have a look at how we would answer this particular question. So if you pause the screen now, read through the question, and I'll explain the answer. Okay, so let's have a look. Let's kind of think of what the question is actually getting at. So one of the key things to focus on initially is to go for looking at the keywords in the question. So we've got the term up here. Okay, he says trying to draw on the ball. We've got the term the importance of meiosis in life cycles of organisms which reproduce sexually. Okay, so it wants to know why it's important. It's worth 3 marks, so we should be thinking about kind of 3 things that we can say. Okay, so I'm just going to bullet point my response. This will look at the importance of meiosis in the life cycle of organisms. Easy thing to say about, first of all, is it's going to produce haploid cells. Okay. Okay, so we've got producing haploid cells. That's the initial first thing. So if we've got haploid cells, so we've gone from diploid to haploid. Next thing to think about is, well, why is that important? So that on fertilization we can return to our diploid number. Okay, so it can ensure that we can maintain the chromosome number in each generation. Okay, so maintain the chromosome number when those haploid cells fuse. Okay, so we can kind of get back to that diploid number after sexual reproduction has occurred. Why else is it important? This is the key thing. It's going to introduce that idea of genetic variation. And if you're going to mention genetic variation, it's always mentioning how that occurs. So you could talk about crossing over. Okay, and you could also talk about independent assortment. Okay, so the three things we're looking for: producing haploid cells, we can maintain the chromosome number from when fertilization occurs, and it's going to introduce genetic variation. Keyword there's the term variation. Okay, here's that example of looking at applying what you know to something you're familiar with. So we're looking at the life cycle of a moss plant. We can kind of go through and just kind of follow through what the process is. So we've got an embryo here that is diploid. It's going to produce a spore-producing moss plant. Okay, so a multicellular diploid plant that can produce spores that have N. So the key thing to look for here is I've got N, and I've got 2N. So I'm kind of going from 2N to N, like so. This spore can go on and produce a gamete-producing moss plant. I've got male and female gametes. They can fuse to produce our zygote. That can produce our embryo. That's 2N again. So we kind of always look out for the numbers 2N, 2N, N. And it says on the diagram mark with an M where meiosis takes place. I've got to put an M somewhere here. Well, with the meiosis, it's about going from a diploid cell to a haploid cell. So if I'm looking at my 2Ns and Ns, if I'm going from 2N to 2N, okay, I'm maintaining the chromosome number. I've gone here from 2N to N, I'm halving that chromosome number. So I need to put an M in that location, okay, and that's where meiosis has occurred because I've gone from diploid down to haploid. Okay, and that should be how you answer that kind of question. Okay, I hope you found that useful. Let's say any questions, drop me an email and I'll try and respond to help you go through quite a complex process. Okay, I'll also make sure you answer the questions that I've given you on your email. Okay, see you soon.